Respected moderate, thanks for your kind words. Respected faculties, research employees and participants who have joined offline and online. Today we will discuss zoonosis and lab animals, challenges and way forward. So every year on 6th of July, we celebrate this day in honor of French biologist Louis Pasteur, who was really a great scientist, and he has introduced anti-rabies vaccine on the day 6 July 1885. Zoonosis is a Greek word made up of two components, zoon and nozoon, which means animal and disease. And this term was introduced by Rudolf Virchow. As per WHO, zoonosis are those diseases and infections which are naturally transmitted between vertebrate animals and man. So initially it was thought that zoonotic diseases has jumped from animal to human. CDC further elaborated its etiology and other aspects, and it could be viral, bacterial, parasitic, fungal, etc., etc. Now let me understand the mode of transmission, which is really very important. It could be direct contact with saliva, blood, urine, mucus, feces of infected animal, as well as petting and touching, and bite and scratches. So if we are dealing with animals, we should be aware of these different mode of transmissions. Another mode could be indirect, where we get infected while roaming in areas where animals live and roam, and also get infected by coming in contact with ancillary objects like aquarium, pet habitats, barns, pet food, water dishes, etc. Another mode could be vector borne, and here we are being bitten by the ticks, insects, mosquitoes, etc. So they serve as the vector, means like postman. Another mode could be the foodborne, where either we are eating or coming in contact with contaminated food, and kindly note unpasteurized milk, undercooked meat, egg, raw fruits and vegetables. Raw food was very highlighted during spreading of Nipah virus. And the last one is waterborne. Here either we drink or come in contact with the contaminated water. Regarding epidemiology, CDC has highlighted that about 60% of infectious disease have spread through animal, means zoonotic in nature. And more dangerous scenario is about 75% of infectious diseases are somewhere of animal origin. So based on transmission, we can divide zoonosis in different subsegments. Anthropozoonosis, where disease comes from animal to human, brucella, rabies. This list is endless. Another mode could be zooanthroponosis, where disease is being transmitted from human to animal. And tuberculosis is very alarming disease here. Then amphigenosis, where disease may travel in either direction. And very important, eugenosis, where human is obligatory host, about which we learned in our introductory lecture by Aditya sir. This list is very endless, so let me get in very brief. Zoonotic disease could be bacterial, viral, parasitic, fungal, rickettsial, chlamydial, protozoal, and very importantly, a cellular non-viral pathogenic agents, which is called prion. It causes mad cow disease in animal and CJ disease in human. Now coming to the next portion, the prevention and control of zoonotic diseases. In very short, there are 4 types of surveillance which is practiced: pathogen surveillance, then sero surveillance, it is community surveillance. Third is very important, syndrome surveillance. We got some set of symptoms through huge data analysis, which help us to detect early symptoms based on which we can diagnose any zoonotic disease, and the risk surveillance that you know very well. At personal level, personal hygiene management and personal protective equipments help us a lot to fight against zoonotic diseases. Proper disinfection and sanitation, and kindly remember proper disposal of general and bio waste. This waste is very potential source of zoonotic diseases. And last but not the least, continuous education and training. And for control, we have to treat the individual as well as mass vaccination drive helps us a lot. And kindly remember, vaccination is always an economic option in comparison to individual treatment. Now coming to the laboratory animals. Laboratory animals are exclusively bred and allotted or traded for experimentation purpose only. And the biggest challenge and the key success of laboratory animal house management is to maintain entire animal colony without any treatment. And how we achieve this? We maintain control environment where light, air, sound, everything, room temperature, humidity, everything is well managed, and strict sanitation protocols. And these all measures make your laboratory animal safe of any zoonotic diseases itself and their transmission. In spite this, there could be 2 breaches. One is zooanthroponosis, where either animal handler or experimenter may transmit disease from human to those animals. And the second is exposure of wild counterpart of that particular animal species. So in the last, let me understand how we can control and prevent spread of zoonotic diseases in laboratory animal facilities. And it is very important because we are almost 100% sure that there is no disease occurrence, no disease transmission through the laboratory animal. So we have to maintain very high standard for maintaining that facility and not getting infected from animal or not giving any infection to the animal. So first and foremost is, which is very important, hand wash at start and end. Many times, mostly we wash our hand at the end, but we forget to wash at start. Then donning and doffing, and kindly remember, it must be in correct order. हम randomly ऐसा नहीं कि कभी ग्लव्स पहन लिया, कभी मास्क पहन लिया, कभी गाउन पहन लिया, कभी कुछ भी उतार दिया, नहीं. There must be a proper order, because our PPE itself could be potential source of infection to us, to our lab, to our community. शू कवर पहन के लैब से बाहर निकल के रोड पे भी रोम करते हुए लोग दिखते हैं. We need to prevent it. So here is the correct order: donning and doffing, And please do not eat and drink inside the animal room and inside the animal lab. Don't sit on the platforms where animal experimentations are going on, or any animal instruments, or any animal belongings, racks, cages, etc. And please don't offer any gathering of family and friends, relatives to the animal facility, and it's not the zoo. So please don't bring or don't call children to the animal facility. Get vaccinated before start of work. Please remember, get vaccinated before start of work. So if we are going to deal with the Macau facility, we need to have the rabies and hepatitis B vaccination. So it depends on the of what type of work you are going to be involved with, and get trained. It's really very important. We are regularly running the training courses under 0102 programs. So whoever is willing and wishing to be part of animal experimentation, it's open for all. You can join there. And even after that, you got the bite and scratches. And actually it happens due to poor handling. It happens due to poor handling. Rat कभी भी अपना hand आपके सामने नहीं करेगा कि आप blood निकाल लो. आपको इवन अगर केज भी चेंज करना है, यू हैव टू पिक द एनिमल. वो खुद से नहीं जाएगा कि बेटे उस वाले केज में चले जाओ, हम तुम्हारा खाना पानी पहुंचा रहे हैं. यू हैव टू पिक, होल्ड एंड ट्रांसफर द एनिमल. सो ऑलवेज there is only chance of bite and scratches if we had developed the poor handling methods. But if unfortunately it happens, it may develop secondary infections, which are not basically zoonotic, but you may develop secondary infections. And secondary infections is of really very great concern. So we should be aware of it. Immediately report it to your mentor, to your animal house in charge, to your animal house veterinarian, and wash very properly with water, soap, and antiseptics, and seek the medical consultation. And at the last, it is suggested and requested, kindly go through the One Health program, which is the need of time across world. It's a global requirement, and it is specifically required for food safety, zoonosis, and antibiotic resistance. Thank you so much.